We will talk about Galaxy. I will introduce you to the Galaxy training network. And the goal is to show you how Galaxy works, how to navigate, that you will be able to start your first own data analysis. First, please register on usegalaxy.eu. It's completely free of charge. It is an openly funded project. You will need that later for the hands-on part. I like to start with this quote: your primary collaborator is yourself 6 months from now, and your past self doesn't answer any emails. Actually, when we think about what we had for lunch, for example, 1 week ago, you usually have to think a lot about that. It's similar with our analysis, data management, and lab work, for example. We tend to think that it's easy to remember everything, but usually we forget things. That's why documentation is crucial. Galaxy project is actually a project that helps you with that. To use their own words, Galaxy is a free open source system for analyzing data, authoring workflows, training and education, publishing tools, managing infrastructure, and more. The aim of the Galaxy project is to make data-intensive analysis accessible for everyone. Everything you will find is open source under the academic free license. It is a web platform, and it was developed at the Penn State, Johns Hopkins, and some other institutions 20 or 25 years ago. But right now, it's a really big community project. The core values are first, the accessibility. Users without programming experiences can easily upload and retrieve data, run complex tools and analysis, use the workflows, and visualize data. Another part of the accessibility is that you don't need fancy computing resources because Galaxy can give that to you. The cool thing is that it captures all the information so that any user can understand that and repeat the whole workflow, the whole analysis they did. Users can share or publish their analysis. For example, if you have any publication, you can share your workflow in Galaxy. The main server is the usegalaxy.org. It is located in the United States. We have the European Galaxy server we will use today that is funded by the European Union, by the Federal Ministry of Research, Technology and Space, by the GFG, and by a lot of different other organizations. A lot of university contributes to that all over Europe. Then we have another server, the French one, and the Australian. The cool thing is we can use all these servers. So if we like to, we can switch. We cannot migrate our data, but you can use that and it's reliable, even if it's a community project. I will give you an overview about the functions in Galaxy. First, we have a history. There, all the data sets that are produced by tools we used are collected, as well as all the operations performed on the data, that you can always go back, you can always see what you did exactly and repeat everything if you want to or have to. Then we have workflows. If we have a cool analysis, we can put that in a workflow to rerun the same analysis on different input data sets. Quite often we do similar analysis again and again, and therefore it is time saving to use these workflows because we can just click on one single button and everything will run, and we don't have to care for anything else. Therefore, we will make use of the job scheduling with these workflows. All the jobs in your workflow will be scheduled just by pushing the run button. For example, if the analysis will take maybe 2 days, depending on your data set and what you do, you can just click on the button that you want to have a notification on your email, and then you can come back in 2 days for your ready analyzed data sets. You can visualize your data. You don't have to download your data, but you can actually use that in Galaxy as well, and you don't need additional resources. Then we have the Galaxy Toolchat. That's a free app store for Galaxy. In March, we had 10,400 tools in the App Store ready to install. It's really easy to do. The tools in there are open source. Our history is the location of all of your analysis. All the data sets that are produced, all the operations performed on the data. For each data set, you get different information in there. The usual ones, the name, the format, size, the creation time, data type specific metadata. Then you have all the different tool IDs, even a version control. You don't have to take care about noting down all the different versions of different tools. It's in there. All the input data sets, parameters you've used, the standard output and error messages. What is really nice, it's color coded. On the example on the right side, it is green. The job was a success. Everything is fine. You can be happy about that. If it's gray, it's still waiting to run. If it's yellow, or I think a little bit more orange, actually, it is at the moment running. And if it turns red, it failed. Additionally, you can't delete your dataset by just one click. So you can't accidentally delete it. But if you click on the delete button, it will be hidden. And then you can unhide the dataset and purge it, and then it will be gone. But still, the parameters of the dataset will be saved so that you can't accidentally delete anything. You can always retrieve that if you just deleted it. We can import different data types. And what is sometimes a bit confusing in Galaxy, we often have multiple ways to do the same things. For the importing, we can for sure copy or paste some text. We can upload files from our local computer. We can upload data from an URL. That's what we will do later. We can even upload data from databases or import from shared data like libraries, histories, or different pages in Galaxy. The data types are usually automatically detected by Galaxy. Multiple tools can only have specific data types as their input. They will recommend these data files in your analysis automatically. It means that we have to convert data files if they are not useful as an input data set or data type for the specific tool. We already have some reference data uploaded, for example, different reference genomes that are already there so that you don't have to waste your own storage space for that. You can create custom genome builds. New builds can be added by the administrators. If you think something is not that useful, something is missing, you can always ask people who do that. Then we have our workflow editor. As soon as we have analyzed our data, everything has worked, we can extract a workflow from our history. Or if we want to build that manually, we can import even using an existing shared workflow if we want to reproduce the work of some other people. Why should you use workflows? You can, as I said, rerun the same analysis on different input data sets, but you can even change the parameters before rerunning a similar analysis. If you think you have to adjust something in your analysis, you can easily do that. Then you can even create sub-workflows, so they can be really complicated in the end. If you want to, you can share all the workflows for publications. Then we have visualizations. We can use some tools. There are just some examples on the right side. What you always can do if you have any issues because of, for example, personal data, you can install your own Galaxy instance or for your institution. It is quite easy. Everything is on GitHub. There are some companies that offer services with the Galaxy instance for these kind of data. When we are using the Galaxy.eu server, everything is stored on servers of the University of Freiburg. The usegalaxy.org server is located in the United States and funded by the NIH. For example, the start of the Galaxy project was in bioinformatics, so there are usually more tools for bioinformatics installed on the Galaxy servers. But since it lives through the community, it is open for all other science domains as well. We already have some tools for digital humanities to analyze text. There is a tutorial for text mining, for example. We have different tools for computational chemistry, for environmental sciences, and so on. There's really a lot you can do there. I didn't have the data type I wasn't able to use in Galaxy yet. It's really flexible. You can even code in the interactive environments like Jupyter Notebooks or RStudio. Sharing is caring. So you can share everything in Galaxy. You don't have to, as long as you don't share, only the Galaxy admins have a possibility to look into that. They usually won't if you don't ask them to, at least on the European server. You can share, for example, directly using another person's Galaxy account and share that with their email address. Then they will get access on the server to your history or workflow. You can use the web link that anybody who has this link can access that, or you can publish on the Galaxy server where everybody can access that from shared data. There's a support forum where you can always ask for help if you need something or want to. Community curated documentation is in the Galaxy Community Hub. We have events all around the world, and there are different trainings made by the community for scientists, for developers, even for the admins. There is a training how to install your own Galaxy instance, for example. And for instructors, there's even a training about how to do training with Galaxy. Everything you will find in the Galaxy training network, everybody can easily contribute to that. For example, I did multiple Galaxy introductions yet, and 2 of them were advertised on the Galaxy server, for example, and now I'm listed as one of the contributors just for doing Galaxy trainings. But it's really nice and it's open, and I really like this community and this style of working. There are different community groups that focus on different analysis. For example, there's a Galaxy climate tutorial group, there's microgalaxy, some that focus on environmental sciences and climate change. You can use different public servers where they have only installed the tools they need so that you don't get confused because of too many tools. What you can see here is the usegalaxy.eu server. You can find all the information, for example, news, different events that will happen, more information about the European server, different projects. Everything is linked there that you can use or that you might need. On the left side, you can find different operations you can do. For example, the tool button where you can search for different tools you want to use. You can find your workflows, visualizations, different histories. There's a lot to explore. This can sometimes be a bit confusing because in Galaxy, we usually have multiple ways to do the same thing. But I will show you. And on the right side is your history. This is some history of another workshop I did. There you can create, for example, new histories. You can switch between different histories and so on. Something important, on the top right side, you can see your data quote. At the moment, I have 1.2 terabytes. You should have 250 gigabytes. When we click on our username, then we go to the preferences. We can change our preferred Galaxy storage, and we can manage different kinds of storage. Our preferred Galaxy storage right now is the long-term storage, but there's another short-term storage where we have 2 terabytes, but everything in there will be deleted after 60 days. It's quite nice for testing a workflow, for trying a new analysis, but you have to keep in mind that everything will be deleted after 60 days so that you download anything important. You can link different clouds for that. For example, your Nextcloud. I hope you've all created an account on the Galaxy EU server and opened the tutorial. This is the Galaxy Basics for Everyone. At the top, there is a little head symbol. There you can see Galaxy training materials as well. Click on that, then go to the Introduction to Galaxy Analysis, scroll a little bit, and there is the Galaxy Basics for Everyone. I will show you everything quite slow and you can follow along. First, we go to the right side on the history, and there we can create a new history. It is now named Unnamed History. We want to change that because otherwise it will be hard to find that again. I will just use the date, the 27th of November, and Galaxy intro. We can add some more information if we want to, or we can even add some tags. There's some recommended ones. Maybe I will just use the data tag, then we can save that. Now we want to have our data we want to analyze, right? We can go to the Galaxy training material. If you scroll a little bit down, you will come to the part upload the iris dataset. And here's the WiNoDa link. We will just click on that on the copy button. As soon as we copied the link, we go to the left side to upload data. Here we can choose on the bottom part, paste and fetch data. Put the link in there. Click on start. As soon as it's green, we can close this view. Now on the right side, we have this grayish field iris.csv. That means that the uploading of the data is scheduled. Wait a little bit, then go to the introduction to Galaxy analysis. We choose the Galaxy Basics for Everyone tutorial. Scroll down to upload the iris dataset, copy the link over here on the right side, go back and upload data, click on paste and fetch data on the bottom, put in our link and start uploading. If it's green, you can close this view. Now, mine is uploaded right now. And as you can see, it is already detected as a CSV file, which is good because it is a CSV file. And we can see some lines of our dataset already in here. If we want to see more of our dataset, we click on the little eye at the right top part of this iris.cv box. We can view our dataset. We already have a preview, scroll through it. In this preview, we can decide to visualize our data, but we won't do that yet. Go on details, click on edit. If our dataset is not a CSV file at the moment, we could change that here, but it is. So we are fine with that. What is quite nice, we have a color coding since we use the data tag and the history as well. It has the same color. It makes it easier to later find anything you need. Now we have to pre-process because we later require some tab-separated input data. And now we have comma-separated input data. We have to change that and we have to remove the header because the tools later will need that they are removed. The file is a CSV file, but now we want to convert that and we need to have a tabular file. Go to this little pencil, edit attributes. Click on that, you will get the same view I already have here. Go beneath edit on data types, assign a new data type or convert that. We want to have the tabular file, as I said, so we don't have to change anything. It's already in here. Create this data set. It now has produced a new data set. The header has changed. If we don't use a hashtag for our tags, it will not migrate. But if we put a hashtag before that, it will migrate. For example, if we have an analysis about a microbiome of mice and we have some mice DNA and some of the bacteria, we can see later on if our data sets are from the mice or from the bacteria. Already in this view, it is color-coded, easily accessible, and so on. You can always add tags afterwards, but it won't migrate back. If you add a tag later in your analysis, only the following produced datasets will get that. My dataset is now converted to tabular. We can view that. We see a nice table again, and we can add tags. Use the hashtag iris. It should migrate to everything we will do in a second. Another thing, the names of the datasets are usually automatically assigned. Here it's not the case yet, but usually in the name will be the name of the tool you've used to create this dataset. This can be confusing, so it's always nice to rename things if you want to find anything again easily. Now click on the pencil icon again, edit attributes, rename, and we use iris tabular as a name. And then scroll a little bit down and save that. Now it has already changed on the right side. Now we want to remove the beginning of the dataset. If you look into the dataset, you see two different headers. On the top, there's another header where the column numbers are written. This is a function of Galaxy to make the overview a little bit better, but this isn't actually in our dataset itself. It's just the view in Galaxy. And what we want to remove is these sepal length, width, and so on. Either go on the Galaxy training material, scroll down, and click on remove beginning, and then it will open. Or click on the left side on tools. Here I can search for tools. If I put in remove beginning, it's already here, remove beginning of a file. Click on that and it will be shown here. We already have detected our data set, so we don't have to take care for that at the moment. It's already the right data set that is added here. Everything is fine, and we can just click on Run Tool. Now it's scheduled, and we have our iris hashtag migrated to that. We have usually additional options. We can get, for example, the email notification as soon as the job is complete, that you will get an email, which is nice for longer workflows. We can reuse it. On the bottom, you can find the help part. If you have a fancy new analysis you're doing, you have a tool you want to use, but you don't know how, you can see if there are already some tutorials. I've removed the beginning on dataset 2, and this is now the name of our new dataset. This can be quite confusing and quite annoying because it doesn't give us too much information about our goal or what we are doing. Go on Edit Attributes again on the little pencil icon. Change the name, call that iris clean. Now we want to know how many species do we have in this data set. For sure, we can count, but we have 150 lines. Could be annoying depending on the analysis we are doing. I don't want to count that. We will try to do that in another way. First, we use the cut tool. You can either go again on the Galaxy training material or search for that in the tools. We want to cut just column 5. Recommended here is column 1 and 2 because maybe a lot of people do that, but we changed that to C5 so that we cut our column 5. Our dataset, since we have changed it to a tabular file, is delimited by tabs, so that's fine. It has already detected the right dataset, our iris clean dataset. So I don't have to change that at the moment. Otherwise, you can expand the view and search for the right dataset, then run tool again. As I said, you can always rerun that. To show you a little bit more of the functions, I can click here on Run Job Again. When we click on our dataset we want to reproduce, then we can click on the Run Job Again. It will open the same tool with all the same parameters we've added. Not what was first assigned, the C1 and C2, but the C5 I've added, which is very convenient because here now we can change anything. We could, for example, change that to column 4. We can change the data sets and so on. In this example, this is a really nice analysis, but for more complex versions where we just want to change, for example, 1 parameters of 10 we've added, it is really convenient that we don't have to put everything in there again. We could, for example, add multiple data sets here as well if we want to. Now the job is done, the cut on data. We will call that iris species column and click on save, and it has changed. View into that again. As you see, we have just 1 column left, column 5. Awesome, that's what we want. But we want to know how many species we have. We use the next tool, which is called unique. Click on that. Again, it has already assigned the right dataset, so I don't have to change anything. We don't need any advanced options right now. Just click on Run Tool. Again, rename this file and call that iris species. We already have a preview here. We have 3 species at the moment. Therefore, we can go on with our analysis. As I said, we have quite often multiple ways to do the same thing. Now we are changing the way we have done this. We use the group tool. If we open the group tool, we now have to enter our iris clean dataset, which has assigned the latest iris species dataset, but we want to have the iris clean dataset in this case. Group this by column 5. Everything is fine. Just click on Run Tool. We have the dataset details. What I really like, you can find at the bottom some estimated resources. Everything we do in Galaxy is cost-free, but still it has some costs to run things. Here you can always see the carbon footprint, the estimated price for the server we've used, and so on. I think it's quite nice. So now we've used the group tool. We have 3 different species in our data set. But now we're going to rerun. Click on group again. Click on rerun job group by column 5. Scroll down a little bit. Insert some more operations. We use the type we want to count on column 1. Don't change anything else. Run that again, because what we want to know is how many samples per species do we have actually in our data set. It takes sometimes a little bit, and I know that it can be a little bit annoying, especially for such easy jobs, because it's not a fancy thing we're doing right now. It depends on the traffic on the server. But as I said, with the histories and the rerun and everything, we really save time in the end. But for your own analysis, you can really save time because you don't have to care for version control, for installation of different tools you need. Usually they are already installed. You can rerun everything and it makes it really convenient. Additionally, you don't have to note everything you did. So you don't have to have your own lab book, for example, or wherever you store these notes for your analysis, text file, whatever, everything is already in here. So it really saves time for us. An AI can predict the folding of protein structures, which is quite a big challenge. This is installed to Galaxy as well. For a small protein, it's the dopamine receptor I've decided for. I think it has less than 300 amino acids. It is small, and I've calculated the predicted folding with AlphaFold in here. And it is quite easy to do. Click on Run Job again. You just have to put in your FASTA file. I've downloaded in a database and just clicked on Run. I did nothing else, right? It's really easy. I've got all these things in here. What I really like is that we get a visualization of these predicted structures we can play with. It's an HTML file. We can, for example, scroll into that. We can get a closer view. We can see the 5 top ranked structures. We can change here for which we want to see. For example, we can look for the surface representation. When we click on dataset details and we scroll down, I was quite shocked actually, because first here we have an estimated costs of almost $23 for this one single job. The carbon footprint, it is estimated for more than 900 grams of CO2, which translates here for 1 month of a tree, charging my phone 110 times, or maybe 62 light bulbs running. It's so easy to use these tools. We sometimes tend to forget which resources we're using. I've just put one file into there. I clicked on Run and I did nothing else. It was so easy. It is really accessible for sure. But the costs, I was quite shocked when I looked into that, because I didn't expect that at this point to be this high for just this small little protein. I'm going to delete this job that did not start. Then I can go, as I've shown you earlier, on include deleted. Now it is red because since I've deleted it, the job was cancelled, so it failed. Click on select this job. and then delete that permanently. We still have, if we show only our active data sets, don't see that here anymore. But if we go back to the include deleted, this dataset is still in here and marked as purged. The dataset itself is not stored anymore, and you can't see the preview, but you can still go back and see the operations you did. As we see, we have 50 samples per species. That's what we wanted to know. Rename that again to iris samples per species group. As I've shown you earlier, we have these 3 different species in there. And it's not too important for you, but we have in our dataset the length of the petals and the sepals. These are 2 different parts from the flower of the iris. Now we are going to do analysis of the data. We use the data mesh tool. Here you can see again all the parameters. Just click on that and open it. We want to use the iris tabular file from the beginning. Then we want to group by field, therefore we enter the 5. Then we want to sort that by input, and we want to ignore the case when grouping. Now we're going to put in different operations. First, the mean, then the sample standard deviation on column 1 for each of them. We will repeat that for all the different columns up to column 4. First, again, the mean on column 2 there. Then we can insert another operation, sample standard deviation for column 2 as well. Then again, the mean on column 3 this time. Again, the sample standard deviation on column 3. And same again for column 4. First, the mean on column 4, and then the sample standard deviation. So we want to have the mean and the sample standard deviation for all the columns from 1 to 4, not 5. As soon as you've entered that, you can run this tool again. The job finished. So I'm gonna open that. Now is the question, can we differentiate between the different species? I made a mistake because the header line is not included. I run that again. We can already see some different numbers. We didn't use the first original dataset because we needed a tabular input file for this tool. And the first data file was a CSV file. That's why we had to change that. The petal length, the mean is different. We can already see that there are differences. It seems like the iris setosa is more different than the others are. But we only have numbers at the moment, and it's always nice to have this visualized. That's what we are doing now. We go on Tools again and search for the scatter plot with ggplot2. Then we put in our iris clean dataset. You can see it doesn't have a header anymore because we removed that. We want to plot on the x-axis the column 1 and on the y-axis column 2. Our plot title will be, in this case, the sepal length as a function of the sepal width. And as labels on the x-axis, it's the sepal length. At the y-axis, it's the sepal width in this case. We have to put in some advanced options. There are the data point options. We want to have user-defined point options where we can change the size, and the relative size of the points should be true in this case. Then we want to plot multiple groups in one single plot. The column to differentiate between these different groups is column 5, where we have the namings. and decide for a color scheme, whichever you want to have. At the bottom, in additional output format, we want to have a PDF. Run this tool again. Now we can view our file. I shouldn't have chosen this color scheme. As you can see, it has used a bright yellow. You can see the iris setosa is quite different, while the vesicolor and the virginica are quite similar in this scatter plot as well. The yellow dots are quite similar distributed than the purple ones. That we can easily look into our dataset with just a few clicks and visualize everything. It's quite handy, especially when you're doing your own analysis. We've just used the sepal length and width. And to do that with the petals, click on the Run Job button, put in the different columns, the different namings, and so on. But I want to show you another very important part of Galaxy. Now that we did an analysis, or at least a little part of an analysis, we can convert these to a workflow. And therefore, we want to first clean up our history so that everything we did, that every job that failed will be deleted. I already did that with the one earlier. I didn't do that with the data mesh yet, where I forgot the header. I delete that now. The cool thing is now, when we convert everything to a new workflow, the deleted data sets won't be automatically included. After deleting everything we don't need, we go to the right side to these 3 rows. This is the history option. Go there, select extract workflow. We have all the different jobs within there. On the left side, the tools. On the right side, the history items that were created with these tools. There's a recommended name in the tutorial. I just shortened it a little bit into exploring iris dataset, and then create this workflow. Now, on the left side, right beneath the tools is the workflows button. Click on that and we can see all the different workflows we have. Right on the top is my exploring iris dataset workflow. If anybody shared some workflows with you, you can click on that. And you can even search for public workflows. There you find the workflows from all the tutorials as well. But now I'm going to be exploring iris dataset workflow. As you can see, it is now just the default output. We can edit that if we want to, so we can put more information into that, descriptions, licensing, the creator names, different tags, whatever we want to. We can play around, we can move everything, get a better overview, however we like to. We can frame things and put some color into that. There's a lot of different options. We can even search for new inputs on the left side. There you can, for example, put some text in, different things, colors, float input, integers, data sets, and so on. You can search for different tools you want to input. We don't do that now because we don't need that. Now we want to rerun everything. We are creating a new history. We just use the date again, and then I will call that diamonds because the next data set is about diamonds. Go to the tutorial again. There is at the bottom another dataset link. Upload this dataset similar to before to this new history. Click on Start and then close that. Now we want to run our workflow. So go on Workflow. So here is my Exploring Iris dataset workflow, and there's the Run button I'm choosing. Now it is detected. Go on Run Workflow. And what is really nice, you can see here, you have a view of our workflow, what is starting. As you can see, all the jobs are scheduled there. And now it should just run through. The dataset itself is about diamonds, about different values, different parameters. You need to determine the value of diamonds. We have dataset collections in Galaxy, and we just work with one single dataset yet, but quite often we have a lot of different datasets and we could put them into a collection. For example, here I have a collection with 172 data sets. If I start a tool, I want to analyze all these things, I can just use as an input the dataset collection, and all these 175 files will be analyzed there and will be put in a new collection so that you have a better overview. For example, here I have a list with 8 additional lists. And in each of these 8 lists is around 170 data sets. I have 8 times 170 data sets in this case, which is quite a lot of data. Here we have an easy overview. I just have to use this one single input and it will analyze everything for me when I start an analysis with that. We even have integration of different research data management tools. For example, as a storage space, we can use RSpace or eLab FDW. Those are different electronic lab notebooks. You can integrate that with your Galaxy that you have and download data into the ELN again after your analysis. You've seen how your first analysis works and how histories and workflows work. And now you should be able to start your own first analysis.